University of Oregon Research Associate, herpetologist, and author. Dr. Titus explores the fascinating facts on poisonous amphibians and reptiles of the Willamette Valley and other parts of Oregon. For over 2 decades, Dr. Titus has taught the popular University of Oregon course, Amphibians and Reptiles of Oregon. After all these years, he still thinks salamanders are the coolest animals on the planet. And in addition, for over 2 decades, Tom has worked as a research geneticist at the U of O. His science encompasses human genetic disorders, ecotoxicology, and ancient DNA, and he has authored or co-authored 30 peer-reviewed scientific articles. Tom wrote the regionally popular book Blackberries in July, A Forager's Guide to Inner Peace, and also writes a blog found on his website, tomatitus.com. And his blog is called Rational Creativity in the Natural World. Tom is also a grateful husband, father, grandfather, gardener, fisher, forager, and recent 3-time runner of the Boston Marathon. And with that, let's welcome Tom for tonight's presentation.
So first of all, I'd like to apologize for that trivia quiz.
You should.
I thought you took it after the talk. That was brutal. 8 out of 10, that's amazing. Good job. Yeah, that's terrific. Thanks, Maggie. And thank you all for coming, and a special shout out to my colleagues from the Eugene Natural History Society that made it tonight. I really appreciate you coming. I'm going to have to fire up my laser pointer. Maggie tells me that I have to behave myself, and by behaving myself, they're taking video, and the only way that they can make sure that I stay in the picture is if I stay in front of the microphone. I would much prefer to be out wandering around in the tables. That's my style. That's how I teach, actually. So I won't be doing that. I'll try to stay put. And I just wanted to add a couple of things here on the opener. Don't call me doctor. I go by Tom. The only time I use doctor is when I'm trying to get something done at the Department of Motor Vehicles, you know. And even though I'm in an institute of neuroscience, I don't know a damn thing about neuroscience. I'm really a fish geneticist. They keep the lights and the heat on, okay? But it's been a great place to be, so. So the first thing we need to do tonight is just give you a little intro to amphibians and reptiles. They're 2 very different groups of animals, and it has huge impacts in terms of where you find them and what they're all about. And so let's have some feedback here. How would you describe an amphibian skin? Give me an adjective. Wet. What else? Cool or cold? Slimy, did I hear? Yeah. Yeah. Good job. Yeah. In fact, that's about right. Yeah. Moist, slimy, cool. That really is pretty dead on. And so what I have here is a cross section of amphibian skin, and what you're going to see here is this layer right here is the epidermis, and there's a layer of actually dead keratinized cells right on the outside of the epidermis. Here's the outside of the animal. This is inside, but it's a very thin layer, and it's actually a really terrible barrier to water loss. They lose water like crazy through that thing. So, and then down here in the dermis, this is important because you have an incredible amount of complexity in there. You have glands that make chemicals to attract mates, hedonic glands. You have granular glands that make poisons, which will be the feature of a lot of this talk. And then you also have glands that make slime. They make slime. They're really good at that. So an incredible amount of complexity there, especially in the dermis. But again, this very thin barrier to water loss right here. So just to summarize that, an epidermis with a thin layer of keratinized cells, poor barrier to water loss, and then these specialized glands for making chemicals and toxins and mucus. And by the way, I think what I'm going to do tonight, rather than field questions during the talk, is just power my way through and then take the questions at the end. Otherwise, hopefully I could be up here for an hour and a half, but Maggie doesn't want that, and you might not either. So let's just buzz through this thing, and then you can scribble down questions as I go. So how would you describe a reptile skin? Dry. What else? Scaly. Sorry? They shed it. That's true. They do shed it. Yeah, so do amphibians, actually. You just don't see it happening. But warm, cool, depends, right? Yeah, so dry, scaly, good job, and often warm, but sometimes cool, depending on what the air temperature is, because both these groups of amphibians get their body temperature from outside sources. They can't make and maintain their own internal temperature like you and I can at 98.6 degrees Fahrenheit. So that's great. Here's a cross-section of snake skin. And here's the epidermis right here, and the dermis below, and there's the outside of the animal, inside of the animal. And here's that layer of keratinized cells. It's the same layer of keratinized cells that you see in an amphibian, but it's actually, and I don't have a scale bar up there, but it's much thicker in a reptile, much thicker. And there is that layer of keratin that gets shed off as a snake skin. And you see that often when you're out walking around in nature. What's cool about that are those two scales right there that have to come off of the eyes. So snakes don't blink, right? They don't have eyelids, so they have transparent scales over the eyes, and those have to be shed off. If they don't shed them, the snake ends up blind. So a snake that's shedding actually does have a really milky-looking kind of bluish look to its eye because they're secreting a fluid underneath that scale to make sure that they get rid of it. So there's a garter snake skin. Lizards shed like a sunburn, so you never see that, except for alligator lizards. I don't know why. Alligator lizards shed it all off at once. Most lizards shed like a sunburn. So you have this epidermis with this really thick layer of keratinized skins, which is an excellent barrier to water loss, and very few specialized glands in the dermis. It's boring compared to an amphibian. On the egg side of things, there's an amphibian egg. There's a frog. There's a salamander and a stalk on the end of the salamander that might be used to attach that egg to the inside of a rotten log or a moist place in a rock pile like a talus slope. And so there's the business part of it. That's the ovum that ends up being fertilized and develops into the amphibian later on, or maybe a larva, maybe not. And it turns out that there's no shell on that egg, no shell. All those layers right there are just jelly. So if you take that egg out of the water and put it in a dry place, what happens? It dries out. It dies. It can't make it, okay? So there's a great example. This is the northwestern salamander. There's the adult right there. They're about that long. They're pretty good size, chocolate brown animals. My wife says they look like space monsters. And they kind of do, and it's because of these big parotoid glands right here on the side of the head that have lots of granular glands that make poisons. They secrete those when they're agitated. But the eggs are really neat. You may have seen them in a pond somewhere. They're about the size of a baseball, maybe even your fist, really stiff jelly that connects them all. And they turn green over time because there's a symbiotic algae that grows inside the egg capsules. So no protective shell, dries out really easily. Compare that to a reptile egg. Let's not go through all those parts. There's the embryo right there. Its little legs tell you it's probably a snake, right? Wrong. It's a lizard. Okay, snakes don't have legs, not even as embryos. But there's the important part right there is that shell. And usually it's a leathery shell, except in some turtles will make a calcified shell like a chicken. But it has a shell, and that's pretty important because that makes it resistant to water loss. So a reptile like a snake or a lizard can lay the egg out in the soil somewhere, and it's not completely resistant to desiccation, but they don't have to be laid in the water like an amphibian egg does. So all of this has huge implications for where you find these animals in our part of the world. And here's a pretty typical distribution of an amphibian. There's our northwestern salamander, our space monster right there. And here's its distribution in Oregon. What's that right there? Cascades. They go right up the west flank of the Cascades and they stop because everything after that is dry as a bone, okay? And you see that repeatedly in amphibian distributions. So here's another one. There's a reptile, a Pacific gopher snake. Some of you may have grown up calling that a bull snake. They're not actually. Bull snake is a closely related but different species than the gopher snake. We have them here in the Willamette Valley, but then they're all over the place out here in eastern Oregon. And again, you know, they need water. They don't so much, okay? So that really is the deal. It's really about water, but also about temperature. In the Willamette Valley, we have enough sunny days over the course of the active season, which would be about, say, April, although now it's getting to be kind of like March, okay? But April through about now, October, and they can get enough sunny days to bask and get warm and develop eggs, mate, develop eggs, and lay them, reproduce in the Willamette Valley just as they can in eastern Oregon, so their distributions tend to be very widespread. But that's something that's related to temperature, okay? So, fun facts to know and tell: 29 species of reptiles in Oregon, 31 species of amphibians, give or take. And in the southern Willamette Valley, or in the Willamette Valley, we have 10 snake species, which is the same number as you find in southeastern Oregon. They're slightly different species, but in fact, we have the same snake diversity, if you just count species, as southeastern Oregon does. And the only other thing I would add to that is they aren't mostly garter snakes. We have a lot of different snakes. I don't have time tonight to go through all of them. They're amazing, okay? But in fact, yeah. And the other thing I want to add, because I always do, because it's my sworn duty to do so, eat more bullfrogs. You've got to eat those things, you know. They're terrible. They're invasive. Yes, you can catch them with a fly rod. Yes, you can catch them on a bass outfit. You can catch them any way. You can shoot them. You can do anything, okay? But go ahead and eat them. Don't waste them. Eat them. They're not bad. They're invasive. They're terrible predators in our aquatic systems. Yes, they eat ducklings. They eat garter snakes. They eat pond turtles. They eat anything they can get their prodigious mouth around. Okay, they're amazing predators. So just had to throw that in at the end. Okay, Eye of Newt. Well, I did a bait and switch on the title. Maggie said a Halloween theme. Okay, Eye of Newt. Well, I'll just tell you they're yellow. That's it on the eyes, okay? What we really want to talk about is the skin. That's where all the action is for tonight, all right? That's our tale of toxicity for tonight. So there's our rough-skinned newt right there. This animal is not so rough because it's a male that's been living in an aquatic habitat probably maybe all year long, because the males will sometimes just stay in the pond, and they take on a very smooth appearance. The toes, actually, they start to redevelop webbing around in the toes, and they redevelop a caudal fin, a tail fin. So some people call it reverse metamorphosis, but that's kind of overcooked, overbaked. All right, this isn't really, but they do become more aquatic. There's an animal. Ever seen this? When they're agitated, they'll arch like that into this Unken reflex, and they're showing off that bright orange coloration underneath the tail, and also sometimes they'll raise the chin and show off all that orange coloration. And what's that saying? Don't eat me, or, well, go ahead. You know, you can go ahead and eat me. People see more of these animals. So in terms of salamanders, that's the animal everyone knows. Yes, they are salamanders. They are a subset of salamanders. Newts are a subset of salamanders. But they're out marching around in the middle of the day. How come? Because they can be. They're so toxic that they can be out there in the middle of the daylight, and they don't have much to worry about. There's enough toxin in the skin of one of these animals in our area to kill 10 to 20 adult humans. No joke, okay? Amazing, right? Now, you would have to extract that in its purest form to kill all those people if you had any ideas along those lines. But So again, back to our amphibian skin, there's the granular gland right there. Newts have a bunch of them, that rough, warty skin. All those warts are concentrations of granular glands that are capable of making the toxin that makes them actually so deadly. And in what they make is this stuff. It's called tetrodotoxin, or if it comes from a newt, you can call it terikatoxin, because terika is the scientific name of the rough-skinned newt. Terika granulosa is its scientific name. I almost flunked out of chemistry as an undergrad, so these things don't mean a whole lot to me, but I think they're really cool. Tinker toy diagrams. Importantly, those atoms out here on the outside are charged. There's a charge to that molecule. Sometimes we shortcut the name and call it TTX, okay? But tetrodotoxin is what they're making. Tetrodotoxin, you can buy anything on the internet. Okay, where was I? How did I end up in academics? I could have made a fortune making whiskey flasks with this chemical structure of tetrodotoxin on it. Or a cute little tote to carry your chanterelles in. Or you name it, okay? It's amazing what you can find. So how does that stuff work? There's too much going on in here and it's too small, and I apologize for that, but here's the thumbnail sketch of it. This is a nerve cell right here. Nerve cells, the beginning of a nerve cell usually has a bunch of these projections called dendrites, and then there's a long skinny bit called the axon, okay? What that nerve cell is all about is conducting an impulse down to a muscle fiber. There's a skeletal muscle fiber, and it makes something happen, like run away from the cougar or pick up a beer. Good idea. It's not often I get to drink a beer and give a talk. It makes something happen, and sometimes the neurons are stretched in sequence. There'll be more than one neuron leading to a muscle fiber, but there has to be an impulse propagated along that thing to make that muscle jump, to do what you need it to do. And that happens because sodium ions, and that's the same sodium that you find in table salt, sodium chloride, okay? Once you put that into water, it dissolves, and the chlorides go one way, and the sodiums go another way, and there's all that sodium in solution. And what happens then is the sodium rushes into that nerve cell when it's stimulated and propagates that nerve impulse. That has to happen, okay? Also, potassium has to go out, but that's kind of minor compared to the sodium. How do you make, well, we'll get there. Here's a diagrammatic view. So how do you make a good poison if you're an animal? Well, this is a channel. The fancy name for it is voltage-gated ion channel or sodium channel. That's the channel through which that sodium goes into that nerve cell and propagates the impulse, all right? That's it. And you can see it's plenty big enough for a sodium, and it's hooked to a water here for some reason, but plenty big enough for that to travel through and cause that impulse to be propagated. But there's our tetrodotoxin right there. Notice it's big, and it blocks that channel. And not only is it big, but it has all the right charges around the outside to stick in there and keep the sodium from going in and propagating an impulse. What happens then to your muscles? They don't work. You go into paralysis, okay? You do things like you forget to breathe. That's after your lips go numb, all right? What happens if you pick up a rough-skinned newt with your bare hands? What happens? I'd buy you a beer if I wasn't a poor herpetologist. Nothing happens. You can pick them up with impunity, okay? What happens if you eat a rough-skinned newt? And everyone's going, why didn't he put that on the friggin' trivia quiz, right? I could have done that, yeah. Yeah, you'd die, okay? And in case you're in doubt about that, here's an actual case history. Drank 150 milliliters of whiskey. Already, you know it's going south, in addition to the fact that he's a 29-year-old male with an underdeveloped frontal lobe, okay? At 6 p.m., he'd already had the 150 milliliters of whiskey, He swallowed a 20-centimeter newt, an adult newt, on a dare. Of course you would. Why wouldn't you do that? And he had tingling of the lips. Then after 2 hours, he was numb, he was weak, and he thought he was going to die. So someone said, well, dude, if you think you're going to die, how about we give you a ride to the ambulance? He declined. They left him alone. 15 minutes later, full-on cardiopulmonary arrest, and he was dead. Boom. That's only one, okay? They're not always lethal. Sometimes people don't decline the trip to the hospital in the ambulance, and they get their stomach pumped, and they survive their idiocy. But the only smart thing about his evening was he thought he was going to die, and in fact, he did. Yeah. So what's really amazing is, in case you were actually thinking that you were going to use a newt as a suicide pill, like you think that it's the end of the world's coming and you want to just off yourself because you don't want to go through it, and believe me, I totally understand, all right? You got to know that that stuff doesn't cross the blood-brain boundary, so you are fully conscious while you're going into full paralysis and you're asphyxiating because your breathing apparatus is seized up. So think twice. You know, you might want to like stockpile Nembutal or something like that. I don't know, some other way. This is not a good way to go out, okay? All right, but there are animals on the planet that can eat those things. And drum roll. Thank you. I thought I was going to have to yell for one. Garter snakes can eat newts. And so I have a picture to prove it. There's the garter snake. There's the hapless newt. It's halfway down the garter snake's gullet. That proves it, right? Of course it doesn't prove it. That garter snake could have drank 150 milliliters of whiskey and taken a bet from her friend and been dead 2 hours and 15 minutes later from full-on cardiopulmonary arrest. You can do anything on the internet with photos. But in fact, they can do it, and they can get away with it. And the question is, how do they pull that off? Well, we're back to our cartoon here with those channels where the sodium's supposed to go through, but the tetrodotoxin is getting in the way. Here's what it looks like in a garter snake, okay? This is actually two different versions of a garter snake, a common garter snake sodium channel. And this is sort of a cartoon rendition with the coils shown in the protein. So those are two groups of amino acids that make the protein. There's the channel right there. There's the tetrodotoxin right there. Here's what it really kind of looks like, all sculptured. It's beautiful, I think. It actually looks a lot nicer than those Tinker Toy drawings of chemical structures. Every one of those colored blocks right there is a change in an amino acid, which is the building block of that protein, that sodium channel. Every one of those red places is a change compared to your sodium channels, for example. And what happens is, because of those amino acid changes, remember that tetrodotoxin molecule's charged, it no longer sticks into that sodium channel like it would have without all those changes. And so the snakes become basically immune to the tetrodotoxin. They can't, it doesn't really affect them, okay? It's really cool. And it turns out that you can actually have a subset of those mutations that lead to those, that lead to making that TTX less toxic. And the fewer of those you have, the less resistant you are. And so where the newt, well, we'll get to that. But at any rate, you get the picture. Yeah, garter snakes are amazing. They can eat those things without the whiskey. So there's this really nifty thing called an evolutionary arms race between the rough-skinned newts and the garter snakes. The garter snakes are eating the newts. The rough-skinned newts aren't particularly fond of being eaten. From an evolutionary perspective. And so what happens? Well, maybe not that, but I love the cartoon so much that I had to put it up there. There's the snake with its tetrodotoxin resistant gas mask on right there, and the newt's getting ready to douse it, but you're all sophisticated enough to know that it's just a cartoon and that isn't really what's going on. At any rate, here's a picture. There's way too much going on in there, and I kind of apologize for this, but I couldn't figure out how to parse it. I stole it. Most of these things I've stolen and tried to attribute them off the internet. But This is based on a lot of work, a lot of research. And then over here, you have, this is Western North America, okay? There's the Willamette Valley right there. And what you see is in orange, newt toxicity is high, where it's orange or red. So we have some incredibly toxic newts in our area, very toxic, the most toxic newts in our area. That 10 to 20 dead human newts, right? And so there's prey toxicity, and it varies tremendously. So up here, like up around Vancouver Island, British Columbia, they're not all that toxic at all. Or down here further south into California. So you get the picture, but what's fascinating is you look at the predator resistance, so that would be the garter snakes, right? The predator resistance is high where it's orange and red, and lower where it's green and purple, and you see the same kind of pattern. High newt toxicity, high predator resistance going on. And in fact, all that's been worked out with real data that we're not going to dive into. And then down here, they basically overlaid those two things, and they just said, how much mismatch is there between garter snake resistance and the newt's ability to produce the toxin? And I'll just summarize that briefly by saying not much. There's a tremendous amount of correlation between newt toxicity and garter snake resistance to the tetrodo, to the Tarica toxin. So where they're highly toxic, the newts, the garter snakes are more resistant. Where the rough-skinned newts are less toxic, the garter snakes have lower resistance. And there's this really strong correlation between toxicity and resistance where the ranges of those two animals overlap. And the last little tidbit here is in some places, even though we don't really think of wins and losses in the world of evolutionary biology, the garter snakes are actually winning in some places because they become so resistant to tetrodotoxin that a newt can't possibly produce enough of it to overcome their resistance. Can't do it because the newt's only that big. And interestingly, if you take the tetrodotoxin out of the newt and give it a shot with its own poison, you'll kill it dead, okay? So they have to have it sequestered in those granular glands in that rough skin to carry it around, all right? It's amazing. But at any rate, the snakes are so resistant that the newts are never going to be able to make enough of that stuff to kill them. So they kind of won. Home run. Enough of that. Skin of toad. Speaking of which, I have some props up here. If you ever get a chance, go to my blog and read a piece called Dead on My Dashboard. All these things live on the dashboard of my little red pickup. I have a bunch of stuff up there. There's a rough-skinned newt. I don't know what the shelf life of tetrodotoxin is or Tarica toxin, but it might be expired on that one. It's all dried up and mummified. That's cool. Anyway, you can pass it around if you want. Don't eat it. Oh, there's a lizard mummy, but it's not the real lizard mummy I'm going to talk about later. That's a western fence lizard mummy. Those are the blue bellies that you see all over the place around here. Oh, and since I'm not going to actually talk about wing of bat, I brought a wing of bat just because it's Halloween, okay? And this poor guy, I found him with only one wing and apparently clipped a power line going by and lost a wing, so I put him on my dashboard. Anyway, so there's wing of bat, mummified bat. There you go. Don't eat it. Anyway, back to the toads. Will kissing that toad make you feel like a prince? I don't really. That is a very cool animal. This is the Colorado river toad. It lives down in Arizona and Southern California in the desert Southwest. Actually, they get pretty big, you know, they're like that. And they're protected over their whole range now. They're listed as either sensitive or species of concern in a lot of areas. Notice the warts. No, they don't give you warts. They only have warts. What do you suppose is inside those warts? Granular glands that make poisons, right? But in this case, it's not Tarica toxin, okay? So there's our amphibian skin. There's our granular glands churning out toxins, but these are toads, not newts, all right? They make bufotoxin. Now, bufotoxin is a name, I love it, because bufo is the genus of the toads, the Latin name of the genus for the toads. So it's bufotoxin. It has a lot of different stuff. It's a cocktail of different things. There's epinephrine, you know that one, right? That's what you get. The EMTs give you a shot of that if you need to get your heart started again. Norepinephrine, bufalin, bufothionine, all this stuff. Serotonin, that's a feel-good thing. Now we're getting somewhere, you know. But really the star of the show is this. These animals produce an incredibly high amount of 5-HO-DMT. You know about DMT? How many people have heard of DMT? Yes. DMT is amazing, okay? And so it's this version of the Colorado River toad's DMT is really similar to that DMT that's made by plants and is also synthesized in that DMT that I think you can find in, I can't even remember, mushrooms. But at any rate, let's focus on that stuff, bufotenin or 5-HO-DMT. Oh, there's my chemical structure. I don't know what it means. It's just cool, and it spins in nicely on PowerPoint. It's similar to psilocin, which is made by the whoopie mushrooms. So it's the same stuff and very similar to LSD. And it actually works by binding to some special receptors in these serotonin-sensitive brain cells and kind of makes things happen. Well, here's what a guy named Wade Davis wrote about back in 1991. He actually got some of this stuff out of a Colorado river toad and he smoked it. And he wrote this down, okay? Shortly after inhalation, warm flushing sensation, sense of wonder, well-being, auditory hallucinations, insect cicada sound that ran across my mind, seemed to link my body to the earth. Oh my goodness. I was indoors, but I felt the earth, the dry desert soil passing through my fingers. The stars at midday. Oh man, if I could write like that, I'd go smoke that stuff, you know. That's amazing. I don't believe half of it, but it's an amazing piece of writing. And so why aren't all toads facing extinction in the name of a little harmless chemical stimulation and some shamanic experiences, right? Because that was a shamanic experience if I ever saw one. Man, I'd be down with it. Okay. Well, enter our toad. This is the western toad. Astyanax used to be in the genus Bufo. Boreas, you can recognize them. They have these big parotoid glands, same as you saw on that northwestern salamander, and this yellow dorsal stripe down the middle of the back. And warts that you now all know don't give you warts, but they're full of granular glands that are making bufotoxins, okay? Sorry. I'm gonna need to raise that. Thank you for bringing that, because this is the video feed for or the audio feed for the tape too. So, at any rate, here's normally I'd be up on the tables by now. It's true. After I smoke some of this, yeah. That's good. It'll be fine. I'll just. Oh no, it's not staying put. No, it's good. No, this will be fine. I'll just try to behave. I get kind of excited about this stuff. There's the range of the western toads, practically all over the state. Probably the only reason that hole exists right there is because nobody goes there, honestly, you know. And then you may have seen these schools of black tadpoles up on the high lakes especially. Those are toad poles. Those are toad tadpoles. I could talk for a long time even just about them. They are made in profusion, many of them, and earlier in the summer, about August and into September, they metamorphose, and I don't know if you've ever been up there when those little toadlets are migrating away from the water. You can't walk practically without stepping on them, you know. So they're amazingly prolific. They're doing okay in the state, not everywhere though. Oh, thank you. She brought them on a napkin. I won't eat them either. Oh, the bat wing's still on the lamb. So why not smoke a little toad skin, western toad skin? Now, it's funny, right? But my favorite student project proposal ever for my herpetology class was years ago. And this young woman said, well, you know about Colorado river toads? Actually, no, I don't. Well, you know, they might have been used by indigenous peoples for shamanic stuff. And so western toad's actually really closely related to these things. So for my project, what I want to do is make an extract from the skin of these guys and smoke it and keep a journal of my experiences. And I said, maybe over your dead body, but not mine, okay? You got to find another project. But it was an amazing request. I loved her for it. It's one of my favorite stories ever. So why not? Well, I mean, one thing is that a closely related species might have closely related chemical profiles. That's not an unreasonable thing to say, you know, okay? And so how closely related, we might ask, is the Colorado river toad to our western toad? And it turns out really not all that close, not all that close. Okay, there's a toad tree, or at least a bunch of species. Think of that as a family tree. Things that are close on the tree are closely related. There's our western toad right there, and then all the way around and up here is the Encilius alvarius, the Colorado river toad. So they're not that closely related, really. Maybe in the world of all living things, yes. So other things you might want to consider before you go out and do this. No other toads so far have hallucinogenic levels of 5,8-HODMT. But they do have it, so part of your trivia quiz. They all do have it, but mostly they don't have enough to get whatever shamanic experience you want to incite accomplished. It's not going to happen for you. A lot of them do have high levels of bufogen. Well, that only causes ventricular fibrillation. Or bufalin, that only weakens heart contractions. Or bufotalin, which is also a cardiotoxin. Okay, so you got a lot of bad things that are potentially going to happen with your heart. People studied these on prisoners, of course, back in 1955. Literally gave them injections of this stuff to see what had happened. All kinds of bad stuff happened. And I don't know if anybody died or if anyone's admitting to it, but one woman, Esther Inglis, summed it up this way. The overall effect of toad looking can cause a person to have vivid hallucinations, a racing heart, and muscles too weak to carry their constantly vomiting body to the bathroom. I believe her. I believe her. So she didn't say smoking, though. And in fact, there are different effects that are induced from smoking the stuff than from just licking the skin. She also went on to say, if you want to get high, you might find a better way to do it than licking the skin off an animal that spends its life submerged in bacterial-ridden swamp water that's half alligator pee. So leave the toads be. That's my bottom line. I can't do a talk on toxicity without talking about rattlesnakes. This is our northern Pacific rattlesnake. It turns out it is not the only venomous snake in the Willamette Valley. Garter snakes actually produce some venom, and so do I'll just pat myself on the back, beautiful picture of that ringneck snake with the orange collar and everything. They actually produce a venom, but it's really in small amounts, so it was kind of a trick question. It's the only dangerously venomous snake in the Willamette Valley. And garter snakes, garter snakes will produce a mild venom. Well, I shouldn't say it's a mild venom, but it's in very small quantities. Yeah. So yes, we do have them in the Willamette Valley. How many have seen them around here? Around here. Yeah, fair number of you. Yeah. So you do find them mostly in the foothills, eastern and western foothills, but they get down onto the valley floor occasionally as well. And so if you have a snake showing up that you don't want around and you want it out of there, I say, call me, and I'll come down and move it for you. Don't whack it. I'll move it, okay? So this snake actually turned up on Spencer Butte when they were building the new trail up to the summit, and a guy at the city of Eugene knew me, which is not always a good thing. And he sent me an email and said, do you have any suggestions about what we could do with this snake? It's been camped out there for 3 days, and my guys don't really want to work around it. I said, you want me to come and move that snake for you? I thought you'd never ask. So I charged up Spencer Butte, and there was no snake there by the time I got there because we'd had a warm day before. All the days previous to that were cool, and the snake was just camped out, but then it warmed up and it moved. The guys up there didn't kill it, which was terrific, you know. So I gave them my phone number. I went back down, changed out of my poison oak clothes, getting ready to go to work. My phone's ringing. Of course my phone's ringing. Of course it is. And they said, the snake's out. Okay, charge back up to the top of Spencer Butte. I do run, so it was okay. And I caught the thing and put it in a bag and moved it over to the other side of the hill. So it had a good, happy ending. But at any rate, how a rattlesnake works, there's a venom gland right there. That's that big yellow thing. There's a muscle right here that contracts and squeezes that venom out through a duct and into a hollow, kind of hypodermic type fang. That is retractable, so it can fold up into the roof of the mouth when the snake doesn't have its mouth open. And it's just a very effective system. What is that all about? The venom has hemolytic and neurotoxic components. Too much science. Hemolytic that means your cells explode. Okay, so it causes a lot of tissue damage. Neurotoxic means it poisons your neurons and makes you, again, do things like forget to breathe, all right? And it turns out our northern Pacific rattlesnake is mostly hemolytic, and actually that makes them less dangerous than an animal with a lot of neurotoxin, like a diamondback from the southwest, or like the Mojave rattlesnake, also from the southwest, have a lot of neurotoxin in their venom. And juveniles are more toxic than adults. That's interesting. I thought that was a load of baloney until I actually found the data just this past year. So they also have these really amazing heat-sensitive pits. It doesn't show up really well here, but there's one there and one over on the other side. They're on the front of the head, and so it gives them a 3D thermal perception. So what a snake sees when it's looking with its heat sensing system of a mouse looks a lot like that right there. Okay, that's actually an image that was captured from a snake brain. It's amazing. So yeah, so all these things, you know, rattlesnakes, they can detect and immobilize prey. So that venom is, that heat sensing system is about detecting prey. The venom's about immobilizing it, and tracking happens because of that tongue flicking. And they're following the chemicals trail of the animal that they just struck, and now they're waiting for it to die. Then they'll go trail it and find it. And so defense against potential predators of the rattlesnake, that is what we always think about. Oh God, they're going to bite. That is it's so secondary to what this whole system has evolved to do. It's not that. Our snakes are very timid animals. They want nothing to do with you almost ever. And just to drive the point home, that snake that I caught up there on Spencer Butte, I had to wait for it to finish eating this deer mouse right here when I got there. So that was interesting. So they are in decline in the Willamette Valley. They're suffering from a lot of houses, a lot of roads. They're suffering from persecution. You know, the only good snake's a dead snake, and especially a rattlesnake. And they're suffering because of this landscape effect, where because of fire control, a lot of Douglas firs growing up through all that oak savanna woodland that used to be available. That was good rattlesnake habitat, but it's not anymore. It's becoming shaded, and it's shading off their den sites as well. So they actually are listed finally as a sensitive species by the Oregon Department of Fish and Wildlife, which I think is a major step forward, you know. Who knew that a rattlesnake could actually be a species of concern? But they are here in the Willamette Valley. Whoops, that's not what I wanted. And you know who to call, right? I can say that now because it's almost winter, and you'll forget who I was in the next 3 months. So by spring. So every Halloween celebration needs a good mummy story, so I'll just do a quick one. There's me. That's after I've had my coffee, and that's before I have my coffee. I really need the stuff, and you need it for me too. You need me to have it, let's put it that way. There's a sagebrush lizard. That's an animal that lives in eastern Oregon. And there's a sagebrush, there's one in my hands, and there's a sagebrush lizard that's 12,200 years old from Paisley Caves. Amazing preservation. Dennis Jenkins handed me that thing, and I looked at it, and I went, oh, that's a sagebrush lizard. Oh, it was only there at the end of the Pleistocene, you know. It's incredible, okay? So there's Paisley Caves down in south central Oregon, and there's a picture of it. There's actually 7 different caves along that fault. And there's Dennis being happy in front of one of his caves. Dennis spearheaded all the archaeological work that's been done at Paisley Caves, which is amazing. It's been going on for over 15, 16 years now. And what they found is that it's the oldest occupation by humans in Western North America. That's going to turn out to change, and I'm not going to steal anybody's fire, but it's going to change. Also work done at the U of O. It was excavated excavated with modern archaeological methods, and they really radiocarbon dated it extensively. I mean, it's an amazing set of radiocarbon data, and it goes back 14,800 years. People were in that cave. That's a human sample that was a fecal sample, actually, a coprolite it's called, that's that old. But then back to our mummy, there's that sagebrush lizard. It's a long old story how I got into this project, and I don't have time to tell you, but I wish I did. It's actually in there. And my talk's going over, so I won't be able to read it to you, but you can take a look at it in Oregon Quarterly magazine. How do you do that? Well, honestly, I don't know. I'm no good at this stuff. I mean, my deal, I like animals when they're fresh, living, and like I like to work with them right after they're dead. Yeah, look, it's tough because DNA degrades over time. It becomes more and more fragmented, and it becomes chemically modified in ways that make it difficult to work with. And 12,200 years, that's a long time, okay? On top of that, ancient DNA really is best preserved in big bones. Teeth are really great. Lizards don't have big bones, and they have itsy bitsy little teeth. So we're shoveling, you know what, against the tide here. And then contamination, there's only a very small percentage of DNA that you get from a specimen like that actually is from that specimen. All the rest of it is fungal, it's bacterial, it's wood rat pee, it's humans that have touched it or handled it, all of that. So it makes it tough to really work with. So we're getting some expert help with the ancient DNA. I'm trying to put together a trip to Copenhagen to do some work in a lab that does that stuff. And meantime, we've sequenced the entire genome of the modern sagebrush lizard, which still is found about 6 miles from Paisley Caves. So how do you do that? You go out there in the sagebrush country and you noose a lizard. And you literally do noose a lizard. So there's a little sagebrush lizard with a noose around its neck. And then you kind of borrow a piece of its tail. Ouch, but they actually lose their tails. About 50% of animals in any population have only part of their tail. It comes off very easily, and they grow it back. So I'm just trying to make myself feel a little better here for taking its tail. But I took a piece of its tail home and got some DNA out of it, and we sequenced it. And we did a nice job, and I can tell you about that in a whole nother talk. And eventually what we need to be able to do is match the mummy DNA to the modern DNA to find out that we're actually what sequences that we get out of the mummy are actual lizard sequences and not some fungus or bacteria or other contaminating thing. And so it might look like that might be our modern lizard sequence. We might see that in the mummy with maybe just a base pair or two change in the mummy over time. 12,200 years is a lot of time. And we might see something like that where you have a lot of mismatches and label that as non-lizard DNA. So that's the basic thrust of that project. It's awesome. What good is it? I don't know. It's just a blast, that's what. And I might add, it has been funded largely by donations and volunteer efforts by me and through our sequencing facility. There's no government money supporting this project at all. It's just for the hell of it, okay? And someday you'll get to read about that. And then finally, if I could quit hitting the wrong button, you got to tell Oregon Quarterly about your cool lizard mummy project, and they get a cartoonist to draw this thing. And that is so much like me out there, and he had no idea what I looked like when he made that cartoon. It's absolutely scary, but it's called Lizard Tales. It's in the Quarterly. You know something? I got to stop. Thank you so much for having me. And I'm going to try questions. Yes.
So I've heard a theory and actually ran into a gentleman that was trying to document at higher elevation lakes where the rough-skin newt exists. And his theory was that there's less toxicity at higher elevation. Do you know about that?
I don't. But I bet those data are there. I bet you could actually look at some of those localities. So like, for example, one of the localities that went into those graphs that I was or those pictures that I was showing of the distribution of toxicity, one of those included Lost Lake, which is up high in the Cascades, and we could go and look at that. So I don't know the answer to your question, but I know that it's knowable. How's that? So the question was, are newts more toxic or less toxic, actually less toxic at higher elevations? And I don't know, but I do know that the data are out there. Yes?
So do they actually have a neurotoxin treatment? So if you eat one of these lizards or something, not that I'm planning to, but if you eat one of these lizards, do they have a treatment for it, antitoxin?
Nothing. The best hope is that you get your stomach pumped and that you, oh, sorry. I have to repeat the question. I'm so focused on the question I forget to repeat. So the question was, is there an antidote to the neurotoxin, the Turrica toxin that rough-skinned newts are producing? And the answer is there is none. No. All you can do, you get your stomach pumped, not you. They treat this, they got to get the newt out of there. But whatever's in there is there, and you got to get rid of it. You know, there's nothing that's going to help clear that. And I'm not sure, maybe you know that it's cleared by the liver or the kidneys. I don't know. Next trivia quiz. I hope I don't have to take it. So the question was, there's a lot of variation in garter snake coloration out there, right? And you see it. And the question is there any kind of correlation between the coloration and the resistance of the snake to the newt toxin? Do I have that right? Oh, God, I love that question. So then does that make the snake, is the snake giving off a warning that it's toxic? Oh, man, I love that question. And I don't know any of it. But I'm gonna add a bunch of stuff. So first of all, since I have lousy short-term memory, the last part, do the snakes have warning coloration? Probably not, but it's true that for about a month after a snake eats a rough-skinned newt, it is toxic. It has tetrodotoxin sequestered, and there's enough of it there to kill a predator that would happen to eat the garter snake. So who knows, maybe, it is, but they do. And with regard to the variation in color pattern, I'm just going to add that two species of garter snakes that you see here most commonly are the common garter snake, which was the star of our show here tonight, and the northwestern garter snake. The common garter snake around here almost always looks the same. Beautiful, bright yellow stripe on the back and red checkering on the sides and red on the head. The northwestern garter snakes are tremendously variable, and it turns out I don't know if there's any data that I'm aware of about the resistance of different color morphs to the Tarika toxin, but I do know that different color morphs have different performance abilities. And so the ones that are brightly colored happen to be really good sprinters, for example, because they're brightly colored and they need to get out of there if they're about to be eaten. The more prosaic animals, let's say, that aren't so brightly colored are hiding out and using cryptic coloration, and they don't have nearly the sprint speed. I also wanted to take this opportunity to say there's a cost to the garter snakes to have resistance to the Newt toxin, and it is this. When your sodium channels evolve to the point where the tetrodotoxin, Tarika toxin, doesn't sit down in there, it also becomes apparently less efficient at transmitting sodium through and propagating an impulse, and so the snakes actually, their performance drops off. They aren't as good at sprinting and these other performances that would be beneficial to them when it comes to them getting away from a prey animal. It's fascinating, okay? And so it's not always good to be hugely resistant to the Newt toxin if you don't need to be and you're not eating a lot of rough-skinned newts in your diet. Great questions.
Is the garter snake's resistance to tetrodotoxin related to its resistance to whatever tetrodotoxins it's resistant to?
Sorry, I couldn't hear you. I'm going to try.
Garter snakes eat toads.
Garter snakes can eat toads, yeah.
Nothing much else does.
Uh,
They have special chemistry for that.
I have no idea if they do or not. If they do, it's going to be completely different than what we just saw with the rough-skinned newts and the Tarika toxin, because we're dealing with a whole different class of chemicals that they have to deal with. But it may well be that they have some sort of resistance. I mean, those chemicals, those bufotoxins, those various components that are cardiotoxins, they tend to work by either accelerating or inhibiting the contraction of heart muscles by working on, again, potassium going in or potassium coming out and sodium going in. Okay, so it's entirely possible that you could see some kind of evolution going on that would make them resistant to that stuff. Maybe. I love the question. I don't know the answer. As I tell my students, that'd make a great master's thesis someday. You ought to go do that.
So are there any birds that are birds of prey, for example?
None that anyone knows of. But then birds of prey, it's tough to get an animal care and use. Oh, sorry, birds of prey resistant to Tarika toxin. It's tough to get an animal use permit to go stuffing newts down the throats of golden eagles. That's probably not going to happen for us, but I'm guessing they don't, because they don't eat many of them. Now, herons, they're a possibility. Great blue herons, when they're just out there sucking up stuff, I think someone told me they saw a great blue heron eat a Tarika, but for all I knew, it flapped away and sat up in a tree and keeled over dead in 2 hours. I don't know. I mean, made a huge mistake. But not to my knowledge, they aren't. Phil. Are Japanese people somewhat resistant to tetrodotoxin? No, but they are very good at removing the gland in a blowfish, which is where the tetrodotoxin is produced, getting rid of that thing before they eat it. So they might get a little bit, but I think it's a cultural thing that's allowed them to eat pufferfish. It's fascinating. Tetrodotoxin actually shows up in the blowfish or the pufferfish completely independently from newts. It's evolved twice, and it actually shows up in a species of octopus and a nudibranch, I believe, and a whole host of other animals have evolved resistance have evolved tetrodotoxin accumulation of various sorts. Oh, did I survive it? Maggie gets the last shot.
I was going to ask if you had a favorite bullfrog recipe.
The question was, do I have a favorite bullfrog recipe? Bullfrog étouffée. But here's the thing you got to know about bullfrogs. Before you cook them, you need to you skin them, and you should eat the whole frog, not just the hind legs. That's kind of wasteful, even though they're a horrible invasive predator and should all be killed. But you need to remember to take the frog legs especially and soak them in salt water before you fry them. And what happens if you soak them in salt water before you fry them, it neutralizes the difference in sodium outside the muscle fiber and inside the muscle fiber, so that when you fry the thing, you don't have frog legs kicking like this and splattering hot grease all over you and the range. So just do that for me, okay? And then I don't care what you put on them. It actually is. I had a student write a project paper on this. It was called, the title of the paper I still remember, was the first year I taught, which was 22 years ago, Introduction of North American Bullfrogs, Effects of an Invasive Species and Recipes for Dinner. That was the title of the paper. Next question, Maggie.
That's a good one to end on, but does the age of the newt or the age of the toad determine the concentration of the toxins?
So the question was, to the age of the newt or the toad determine the concentration? So it would, in the sense that in the case of the toads, they have to have fully developed granular glands in their skin. So the tadpoles, not so much, okay? Now in the case of the newts, though, the females, before they lay the eggs, actually give them a really healthy dose of tetrodotoxin before they deposit the eggs. So the eggs are toxic as well. But it turns out, I actually lied on one of the trivia questions. There's another animal, I just don't know if it lives around here. Caddisfly larvae, there's a species of caddisfly larva that has evolved to be able to eat newt eggs. And in fact, they have evolved their own version of resistance to tetrodotoxin. You guys have been great. Oh, man. I was just about out of here. One more. One more. Oh, can the caddisflies sequester it? Like become poisonous themselves? Maybe. I'm open to it. But I'm not sure it's actually been tested. I'm a geneticist, not a toxicologist, for crying out loud. What am I doing up here? It's been fun. Thank you. Oh, and just one more thing. I guess I get to do the raffle drawing, but I am a writer and also, and if you want to support my habit a little bit, I have copies of my book up here, Blackberries in July, A Forager's Field Guide to Inner Peace. Most of it takes place actually right in the Coast Range, right over here in the upper Smith River Valley, so you might be interested in that. They're $10 a piece, and I tell you what, I'll donate 20% to the Watershed Council for anything that you buy.